In the last video, we reviewed how to describe mutations, whether it was at the DNA level, protein, or phenotype level. So now let's look at the repair mechanisms that cells have to try to avoid those mutations. So the first assurance that the cell has to try to avoid mutation or changes in the sequence is during replication. And these are activities that DNA polymerase has. So if we just consider this top strand here as template and this new strand is being synthesized, first, DNA polymerase has nucleotide selection capabilities. So if we consider this top strand the template and the bottom strand the new strand being made, as DNA polymerase is moving along, it allows for the correct base to go in. It's selecting for the correct base to ensure that the sequence is conserved. Now, if the wrong base is incorporated during polymerization, the DNA polymerase can use proofreading activity. So in this case, it would detect a distortion in the helix and then use its exonuclease activity to remove a few back and then try again. And then hopefully this time it is correct. So again, this is a form of mismatch repair during replication. E. coli has a very clever way to repair these mismatches if the polymerase doesn't catch them during proofreading. But this has to happen shortly after this region has been replicated, otherwise it won't work. So what happens is at some point, this consensus sequence GATC is methylated, and this is going to happen sometime after replication. So this would be the template strand that has the A methylated in this GATC sequence at a previous round, after a previous round of replication. Now, the new strand that was replicated, there hasn't been enough time for this adenine to be methylated. So this mark is telling the repair machinery which is the original sequence and which is the mutant. So again, this is a mismatch repair mechanism. So there are going to be a series of proteins that have to hop on and they'll detect the distortion in the helix here, and they'll look for nearby GAT sequence. So maybe it's right down here, maybe it's upstream over here. Now, using that methylation mark, it's going to position itself on the double strand so that it specifically removes that base there. It's going to cut the backbone there and then chew back a bit on either side. Then there will be polymerases that come through and they're able to try again and now have the correct base. Then there'll be a ligase that goes ahead and cleans that up. And so again, this is a form of mismatch repair that happens shortly following replication, only in E. coli. Okay, so then at some point after replication, this strand will be methylated as well. So for the next round of replication, both original template strands will be methylated. Now, again, this is E. coli. There are similar mechanisms that happen in eukaryotes, but it's not this methyl, it's not specifically methyl directed. There are other ways that the repair machinery knows which is the template strand and which is the new strand, which would contain the error. Now let's take a look at the repair that needs to be done when DNA damage occurs sort of after replication and is usually associated with spontaneous chemical modification or some other mutagen. So one example of a chemical modification is deamination. This is where a cytosine is modified to become a uracil. And when this happens, there's again a distortion in the helix, and a series of enzymes will come along and modify this. So the first thing that they do is they remove the base that was attached to the sugar and phosphate, and then other enzymes will come along and chew up the backbone in that area. And it's usually a much bigger area than this, but I'm just going to pretend. uh, and then polymerases, repair polymerases, will come through and fix that up and hopefully incorporate the right bases. Maybe they've got proofreading activity themselves to make sure. Then a ligase will have to seal that backbone. So this is called base excision repair because a specific chemically modified base is being removed. Now, these specific names, mismatch, base excision, nucleotide excision repair aren't that important for us. Um, but generally, what's being detected and how it's repaired is key. So DNA damage can also occur through physical modification. So an example of this would be like UV light. And the way that this works is through a process called nucleotide excision repair. So it's a similar thing that's happening. It's just a different set of proteins that are helping to do the work. Now, the original sequence that I had would not necessarily be damaged by UV, so I have to do a different sequence here to have two thymines next to each other. So what happens is UV light can cause these two thymines to form a dimer. And so there's this like covalent bond here, and that causes a distortion in the helix. And so these two thymines will be linked like that. So nucleotide excisional pair will work almost the same way, just a little different. So instead of there being a cut in the backbone and then a chewing back of the sequence nearby the damage, there are going to be two cuts in the backbone. So we'll just pretend like it's bigger here. And then this whole piece is going to come out. So this whole piece will be removed. And then DNA or repair DNA polymerases will come through and hopefully be able to add in the sequence again. And then again, a ligase would have to, seal the nick. So another type of physical modification that occur is our double-stranded breaks, which require different sets of proteins to repair. So this can happen with like X-ray beams hitting the double strands and breaking those covalent bonds. And there are different ways that the repair can happen. There can either be blunt ligation, where you have blunt ends of double-stranded breaks. So you would have two ends like this, and these ends have to be brought together by the repair machinery. There can also be breaks that create overhangs, and so this is the more common way that these are sealed up. So a series of proteins will bind onto these overhangs here, and they will do work to bring those two pieces together. Now, nucleotide excision repair, base excision repair, mismatch repair, all of these things are considered homology dependent because they depend on that template strand in order to repair the mutation back to what it was supposed to be. Double-stranded breaks are non-homology dependent because they do not use the sequence that is in the original strand. They just try to fix that break. So non-homology dependent pathways can lead to deletions and insertions and other types of mutations, but at least there aren't double-stranded breaks in the DNA. Okay, I think that's it for the repair part of chapter 18.